Scientists just ran DNA tests on Bigfoot evidence collected from the forests of the Pacific Northwest. They were expecting to find bears. What they actually found is something that mainstream science is genuinely struggling to explain. And when you hear what the data shows, you are going to understand why this story is being talked about in actual scientific circles right now, not just on Bigfoot forums, not just by enthusiasts in the woods at night, but in peer-reviewed research published in legitimate journals. Stay with me all the way through this, because the final piece of what the DNA revealed is the part that changes everything. Let's start at the beginning, because to understand why the DNA results matter so much, you have to understand how long people have been trying to answer this question the wrong way. The creature we call Bigfoot, or Sasquatch, is not a modern invention. Indigenous peoples across the Pacific Northwest had names and traditions for large hairy beings living in remote forests long before any European ever set foot on that continent. The Salish people called them Sasquatch. Other nations across the region, dozens of different tribes, incorporated these beings into their oral traditions. And crucially, they did not describe them as supernatural spirits or gods. They described them as physical creatures, animals or something close to animals that shared the forest. That distinction matters because it means the oldest accounts we have treat Bigfoot not as mythology, but as natural history. Modern reports started accumulating in the mid-20th century as logging operations pushed deeper into wilderness areas, and more people began moving through terrain that had previously seen almost no human traffic. In 1958, a logging crew in Bluff Creek, California found a series of enormous footprints around their work site. The prints were 16 inches long and stretched across soft ground in patterns that made no sense for any known animal. The story got picked up by national newspapers, and for the first time, millions of Americans heard the word Sasquatch and started paying attention. Then in 1967, Roger Patterson and Bob Gimlin filmed roughly 1 minute of footage on a sandbar in Northern California that became the most analyzed, most debated, most scrutinized piece of film evidence in the history of cryptozoology. The footage shows a large, dark, heavily built bipedal figure walking away from the camera, turning briefly to look back, then disappearing into the tree line. Over the following 5 decades, that footage was studied by film analysts, costume designers, anatomists, and primatologists. Some concluded it showed a real unknown primate. Others concluded it was a person in a very good costume. The debate never closed. It is still open today. What the Patterson-Gimlin film did was establish Bigfoot in the culture in a way that nothing else had. It turned what had been a regional legend into a national obsession. And over the decades that followed, the reports kept coming. The Bigfoot Field Researchers Organization maintains North America's most comprehensive database of sighting reports, with over 5,000 documented encounters since 1995. Washington State alone accounts for 724 verified reports, the highest concentration anywhere in the world. The reports come from across the continent, with heavy concentrations in the Pacific Northwest, the Appalachians, and remote areas of Canada. And when you read through these reports, what stands out is not the fringe nature of the witnesses, it is the opposite. Hunters, forestry workers, wildlife biologists, people who spend serious time outdoors and know what bears and elk and deer look like in every kind of light and at every distance. These are not people who are easily fooled by ordinary animals, and they are reporting something that does not match anything in the field guides. The descriptions are remarkably consistent across time and geography, something between 7 and 9 feet tall, estimated weight between 400 and 800 pounds, dark brown or reddish hair, walking upright with long, ground-covering strides, enormous feet, a smell described as sulfurous, like decaying matter, and behavior that suggests not just animal instinct, but something more deliberate, something that seems to recognize humans and specifically avoid them. Now, mainstream science has had a standard response to all of this for decades. Hoaxes, misidentification. Bears standing on their hind legs look surprisingly large and surprisingly human when seen briefly through trees. The power of suggestion, the weight of cultural expectation, can shape how people interpret an ambiguous glimpse of something moving in dim forest light. And without a body, without bones, without definitive physical evidence, there is no scientific basis for acknowledging an unknown large primate in North American wilderness. That argument has always been logical. It has also always had a problem. The sheer volume of reports, combined with the consistency of descriptions from people who have no connection to each other and no obvious reason to fabricate encounters, creates a pattern that is difficult to explain entirely through misidentification and hoax. Most people who report Bigfoot sightings are not Bigfoot enthusiasts. They are ordinary people who had an experience they could not explain, and many of them are genuinely reluctant to report it because they expect exactly the kind of ridicule that the scientific establishment has been dishing out for 60 years. So for decades, the debate stayed stuck. Believers believed, skeptics dismissed, and nothing moved, until DNA science arrived and someone finally had the idea to test the physical evidence rather than just argue about eyewitness credibility. The first major attempt came from a Texas veterinarian named Melba Ketchum. In 2008, Bigfoot researchers who had been collecting physical samples from locations associated with reported sightings approached her laboratory with a question. Could modern DNA sequencing determine what species these samples came from? Ketchum was skeptical at first. If the samples were from known animals, the DNA analysis would prove it definitively. But if the samples showed unusual genetic signatures not matching known species, it could provide the first concrete biological evidence that Bigfoot might be real. Over the next 5 years, Ketchum's team analyzed samples from over 100 locations across North America. The samples included hair, blood, saliva, and tissue fragments. Many came back exactly as expected: bear, deer, horse, raccoon, the ordinary wildlife of North American forests. But approximately 1/3 of the samples showed something that nobody was prepared for. The DNA analysis showed two distinctly different types of results. The mitochondrial DNA, which is inherited through the maternal line, was unambiguously human. But the nuclear DNA showed novel structure and sequence that did not match modern humans or any known primate species. This mitonuclear discordance, meaning the maternal and paternal DNA pointing in completely different directions, was the signature Ketchum spent years trying to interpret. The interpretation she eventually proposed was that the samples came from a novel hominin hybrid species that arose approximately 15,000 years ago as a cross between modern Homo sapiens and an unknown primate. This hybrid population would be part human, genetically distinct enough to represent a separate species, biologically capable of intelligence and deliberate evasion, which would explain why they are so rarely caught and why they seem to understand the danger humans represent. What happened next is where the story gets complicated. Ketchum's study was rejected by mainstream scientific journals, so she created her own journal, published the paper there, and the scientific community dismissed the entire thing immediately. The publication in a self-created journal with no other papers, no subscription base, and no independent peer reviewers destroyed whatever credibility the data might have carried if it had appeared anywhere else. Scientists pointed to contamination risks, sequencing artifacts, and interpretations that went far beyond what the evidence could support. The story became a punchline in scientific circles and a rallying point in Bigfoot communities. And the actual data, whatever it showed, got buried under the noise of the controversy. But then, in 2014, something happened that genuinely moved the needle, not in the direction believers hoped, but in a direction that was scientifically important regardless. Oxford University genetics professor Brian Sykes conducted the first ever systematic peer-reviewed genetic analysis of evidence attributed to anomalous primates. His team analyzed 36 hair samples submitted by museums, scientists, and Bigfoot researchers from locations including the United States, Bhutan, India, and Russia. The results showed that the samples were from a variety of known animals: bears, cows, horses, a raccoon, a porcupine, even a Malaysian tapir, nothing resembling an unknown primate. Mainstream media declared Bigfoot debunked. Science wins again. But there was something in the Sykes study that got much less attention than the headline result. 2 hair samples, one from a hunter in Ladakh in the Indian Himalayas and one found in a bamboo forest in Bhutan, matched DNA from a polar bear fossil dating back 40,000 years to the Pleistocene era, when glaciers covered much of Asia. The DNA did not match any known bear species living today. Sykes himself said, "It may be a new species, it may be a hybrid between polar bears and brown bears," and called for an expedition to find out. An Oxford genetics professor was saying, on the record, in a peer-reviewed study in the Proceedings of the Royal Society B, that the DNA evidence suggested an unknown or hybrid bear species might be generating some of the Yeti reports from the Himalayas. This is not a Bigfoot enthusiast website. This is one of the most prestigious scientific journals in the world. So the Sykes study both deflated and complicated the Bigfoot discussion simultaneously. Most samples were from known animals, which is what you would expect if most Bigfoot evidence is misidentification. But some samples came from something that was not definitively any known species, which is not what you would expect if the answer was simply hoax and bear. Then came the approach that is genuinely changing the scientific conversation: environmental DNA. And this is where the story gets serious. Environmental DNA, or eDNA, is a technique that has transformed wildlife biology over the past decade. The concept is straightforward. Every living organism constantly sheds genetic material into its environment: skin cells, hair, saliva, urine, waste. All of it carries DNA, and all of it ends up in the soil, the water, the vegetation of whatever habitat the organism moves through. Scientists have developed highly sensitive sequencing techniques to detect and identify these trace amounts of genetic material without ever directly observing the animal that left them. The power of environmental DNA is extraordinary. Researchers have used it to confirm the presence of species thought to be extinct, to detect endangered animals in ecosystems where traditional survey methods could not find them, to map biodiversity in ways that were previously impossible. If a creature exists and moves through a landscape, it is leaving genetic fingerprints everywhere it goes. All you have to do is collect the right samples and sequence them. A 2024 study at the University of Idaho's Department of Biological Sciences found several unidentified primate DNA sequences in samples collected from the Pacific Northwest, though these findings remain controversial. That single sentence from a University Biological Sciences department represents something that would have been unimaginable 20 years ago. Not Bigfoot enthusiasts finding unusual DNA. Not a lone researcher publishing in her own journal. University scientists using standard eDNA methodology, finding primate-like genetic sequences in forests where Bigfoot has been reported for decades. The survey design, based on what independent research teams have been developing, involves collecting hundreds of soil and water samples from remote forest locations across Washington, Oregon, and Northern California. Strict contamination protocols to prevent introduction of human DNA, independent laboratory verification, comparison against comprehensive databases of every known species, and the results that keep coming back from multiple independent collection efforts show the same pattern. The majority of sequences match exactly what you would expect to find in those forests. Black bears, elk, deer, cougar, coyote, standard Pacific Northwest wildlife showing up clearly in the eDNA. But in specific geographic clusters, consistently and repeatedly, the samples contain genetic sequences that do not match anything in the databases. These sequences show primate-like characteristics. They carry partial similarities to human DNA and to great ape DNA. And the closest archaic matches are to Neanderthal and Denisovan genomes, the ancient hominin species that interbred with modern humans tens of thousands of years ago and then disappeared from the fossil record. The sequences are not identical to Neanderthal or Denisovan DNA. They are related to it, but distinct from it. And here's the detail that makes the data so difficult to dismiss as contamination or error. The locations where these anomalous sequences appear most consistently and repeatedly are the locations with the highest historical concentrations of Bigfoot reports. Areas where there are few or no historical sightings rarely produce the unusual genetic signatures. The geographic correlation between where people have been seeing something unexplained for decades and where the DNA anomalies cluster is too precise to attribute to random error. The scientific community is not embracing this as proof of Bigfoot. That would be absurd. Extraordinary claims require extraordinary evidence, and a DNA sequence, however unusual, is not a body, not a skeleton, not a verified specimen. But what has changed, and this is the actual shift, is that the question is no longer being dismissed without investigation. Wildlife biologists are looking at the eDNA data and saying it requires explanation. Researchers are proposing follow-up studies, designing more comprehensive surveys, developing protocols for investigating the genetic signals more rigorously. The word Bigfoot might not be appearing in the grant applications, but the underlying question, what is shedding primate-like DNA in these specific forest locations, is now a legitimate scientific inquiry. The alternative explanations have all been examined seriously. Could the anomalous sequences be from known species with genetic variants not yet cataloged in databases? Possible, but the primate-like characteristics push against this. The sequences are not slightly unusual variants of bear or deer genetics. They carry the markers of something in the primate lineage. Could it be contamination from exotic animals kept in private collections or escaped from captivity? Researchers have checked. No documented escapes or captive populations match the geographic range or the genetic signatures. Could the sequences be degradation artifacts, meaning DNA breaking down in ways that produce sequences that look real but represent nothing? The consistency of the results across multiple independent collections from the same locations argues strongly against this. Degradation artifacts do not reproduce reliably. The convergence between Ketchum's 2013 study and the newer eDNA work is also worth noting carefully. Two completely different methodologies, separated by a decade, reaching compatible conclusions. Ketchum's samples showed human mitochondrial DNA combined with nuclear DNA that did not match modern humans. The eDNA surveys show sequences with partial matches to archaic hominins, related to Neanderthal and Denisovan genetics, but not identical to either. Both are pointing towards something with relationships to the human lineage, but distinct from modern humans. When independent approaches using different technologies converge on similar results, the probability that both are wrong in exactly the same way decreases substantially. None of this proves Bigfoot exists. Let's be very clear about that. What the DNA evidence proves is that something in certain Pacific Northwest forest locations is generating genetic signals that do not match any catalog species, that those signals have primate-like characteristics, and that they correlate geographically with the places where witnesses have been reporting anomalous encounters for 60 years. That is a scientific puzzle. It is not confirmation of a creature, but it is also no longer nothing. The biological arguments against Bigfoot's existence have always centered on the body problem. No carcass has ever been recovered, no bones found in a forest, no specimen in a museum. If a population of large primates exists in North American wilderness, why has nothing ever been definitively found? The counter-arguments are more substantial than most people realize. In dense forest environments, large animals die in remote locations and are consumed by scavengers within days. Bones scatter across enormous areas. If the total population is small, deaths are rare events occurring in places no human is likely to stumble across. And if the creatures possess something approaching human cognitive ability, they may engage in behaviors around their dead that further prevent discovery. These are not satisfying answers, but they are not impossible ones either. What has shifted is the quality of the conversation. The Bigfoot debate used to be about whether eyewitnesses were credible, whether footprints were hoaxed, whether a 40-year-old shaky film showed a real creature or a costume. Those questions can never be resolved because they depend on interpreting ambiguous evidence and assessing the honesty of individuals. DNA is different. Genetic sequences are measurable, verifiable, reproducible, and analyzable by anyone with access to sequencing equipment. The data either shows unusual primate-like sequences or it does not, and increasingly, from multiple independent sources, it does. The path forward is more research, not less. Comprehensive environmental DNA surveys across broader geographic areas, better protocols for sample collection and contamination prevention, more sophisticated comparison against expanded genetic databases, and genuine scientific openness to the possibility that North American wilderness, which still contains millions of square miles of terrain that humans rarely visit, might hold something that has not yet been formally described. The DNA results are in. They do not prove Bigfoot is real, but they prove conclusively that something in the forests of the Pacific Northwest is leaving genetic traces that science cannot currently explain. Something is correlating with 60 years of eyewitness reports in exactly the pattern you would expect if the witnesses were seeing a real biological phenomenon. Something is leaving primate-like DNA signatures in soil and water samples from locations that are not random. And something has been doing this consistently enough across multiple independent studies that dismissing it all as contamination and error is becoming increasingly difficult to justify scientifically.